Hello, everyone. Welcome to today's broadcast, Advanced Troubleshooting for Spray Drying of Pharmaceuticals. I'm Meg Rivers, the Senior Editor for the Pharmaceutical Technology Group and BioPharm International, and I will be your moderator for today's event. This webcast is presented by Pharmaceutical Technology and sponsored by Catalent. Here is a quick statement from our sponsor. Catalent is a provider of advanced delivery technologies, development, and manufacturing solutions for drugs, biologics, cell and gene therapies, and consumer health products. With more than 85 years serving the industry, Catalent has the proven expertise, technologies, and flexible solutions at the right scale to help ensure successful product development, launch, tech transfer, and reliable global supply. Now, before we begin, let's run through a few quick housekeeping things. This webcast is designed to be interactive. Please feel free to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event, and if you have any problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. Now, without further ado, I'd like to introduce today's speakers, Matt Ling, Thomas Lund, and Carolina Corzo. Matt Ling is the Director of Scientific Services and a member of the site leadership team at Catalent, Nottingham, UK. He has more than 20 years of experience in the pharmaceutical industry, primarily working in large pharmaceutical companies with roles in analytical, formulation development, advanced drug delivery, and project leadership spanning all phases of discovery and development. Thomas Lund is a senior scientist at Catalent's Early Development Center of Excellence in San Diego, California. In his role, he provides technical oversight for spray drying projects from preclinical through GMP stages, as well as supports spray drying technical transfers to other sites within global Catalent network. Thomas has expertise with particle engineering via spray drying and broad experience in formulation and process development for the manufacture of solid oral dosage forms. Carolina Corzo is a senior scientist at the Research Center of Pharmaceutical Engineering, where she oversees activities related to lipid-based formulations, particle engineering, and lab-scale spray drying for oral and inhalation products. Additionally, she provides scientific support to the industrial partners on pre-formulation, formulation, solid state, and stability characterization for drug product development. Thank you all for joining us today, and now I will turn it over to Matt.
Thank you. So for the first part of the presentation today, we'll be talking about troubleshooting of spray-dried dispersions for the development of oral products. So the breakdown of the presentation, we'll start off giving you an overview of the spray drying process. I'll take you through some of the considerations that we have during the early phases of development before my colleague Thomas picks up and covers the later phase scale-up challenges. We'll talk about some of the troubleshooting around common spray drying challenges, and then finish off with a case study. So a quick overview of the spray drying process. So spray drying is a technique that's used to convert a solution of a material into a finely divided powder. And in the pharmaceutical industry, this is often used to create amorphous forms of the API through the spray drying of solutions in organic solvents of drugs and polymers. So the solution is pumped into a spray dryer drying chamber. As it passes through the nozzle, it's finely atomized and then is dried rapidly in a stream of hot gas. The gas is normally, in this setup, recirculated through a closed loop. In the course of the spray drying, the atomized droplets are rapidly dried to give a fine powder, which is collected in a cyclone. Smaller particles that are too fine for collection there are collected in a second chamber, and then the solvents are condensed out from the vapor for use or disposal. An important consideration here, and really the crux of controlling the process, is understanding how the solution is atomized and how quickly it is dried off. So through control of that, we can control the properties of the powder. So thinking about the early phase of development. So in general, in this phase of development, companies are looking to try to achieve suitable exposure of their drug in initially tox studies and subsequently clinical studies. We want to assess the safety of the drug, getting up to potentially quite high doses. We want to be seeing some of the initial signs of efficacy, and often a big driver is to get to the clinic quickly and with limited API. So if we're developing a spray-dried amorphous solid dispersion presentation for the drug in this phase, some of the considerations here. So the SDD is often being used to improve the solubility of a poorly soluble drug, and through that improvement in solubility, it's driving the improvement in bioavailability to help achieve the exposure in tox and in the clinic. In order to do so, we need to be able to identify drug polymer combinations that provide the enhanced dissolution, so that we are getting that improved bioavailability. We also need good physical stability for the drug polymer combination, and ideally we want to be operating at a high drug load. So the higher the drug load, the smaller the dose will be for a higher dose of drug. The amount of API that may be available at this stage can be quite limited, and so if we have API-sparing approaches to developing these spray-dried dispersions, then that's going to be beneficial to the supply of the API, and also can lead to a quicker turnaround of the data and the generation of the product. While we are aiming to achieve these early phase goals quickly and with limited API, we do at the same time need to have an eye to the longer term developability of the SDD based product. So thinking about some of the first steps in the early phase. So initially we need to select the solvent system that we're going to be spray drying from, or at least initially spray drying from. So the initial screening, we'll look at multiple solvents, and we're aiming to get to find solvents that give solubility of the API at around 50 mg per gram of API in solution. So that's around a 5% drug load in the solution. And ideally, we want to have the API stable in solution for a few days to enable long spray drying times at the latter end of development. So if we can achieve that, then we can proceed to spray drying the initial test batches. If we're not able to achieve that solubility with single solvents, then we'll be looking to optimize the solubility of the API, potentially through the use of combined solvents. So from the initial screening, we'll look at a few of the solvents that are showing the best promise, and start to look at combinations of those solvents, potentially 80-20, 20-80 ratios. And then when we find a system that does have acceptable solubility, we can then proceed with the initial spray drying. And some of the common solvents that we'd be looking at here So we're often using alcohols, so methanol, ethanol, isopropanol, often ketones, so acetone, methyl ethyl ketone, methyl isobutyl ketone. Chlorinated solvents such as dichloromethane have a great utility here. They're often very good at solubilizing both the drug and the polymers that we're working with, and then sometimes solvents like ethyl acetate. Key considerations in the solvent selection are thinking about the residual solvent spec that we're going to need to meet for the finished product, which of course is related to the toxicity, the tolerability of the solvent. We also need to consider the compatibility of the solvent with the spray drying equipment. So some of the more aggressive solvents that we may look at to dissolve the API may also be very good at dissolving some of the seals that we have in the spray drying equipment. And of course, the boiling point of the solvent will also be important when we're establishing the spray drying conditions that we're going to be using. So that covers where we go with the solvent. The other part of the solid dispersion is the polymer. So some approaches that we take to determine which polymer we're going to be using. There's broadly two aspects that we need to consider. So the first part, we need a polymer that is going to be stabilizing the amorphous state of the API, because we're normally using the amorphous state of the API to improve solubility to get the bioavailability enhancement. So one of the first parts of the screening is to look at combinations of the drug with different polymers at different drug loadings to see which combinations can produce amorphous films, and then through stressing those on stability, which of those can remain amorphous on storage. The other important aspect is can the polymer maintain the supersaturation of the drug following dissolution. So, in other words, when the solid dispersion releases the drug on ingestion in the intestine, which polymer will maintain the solubility of the drug at an elevated level. And there's a screen that we use routinely here, a liquid-liquid phase separation screen, where we are using fiber optic microdissolution equipment, and in the dissolution medium we have the polymer pre-dissolved at a known level. We then titrate the drug into the dissolution medium, so the drug is dissolved in a water-miscible organic solvent at a high concentration. We titrate the drug in, and through monitoring the UV response and the scatter of the UV trace, we can determine when the drug is going to come out of solution, so either crystallizing or phase separating. And by identifying where that separation occurs, we can determine how much polymer is required to maintain the drug in solution. So from a combination of those two experiments, we can determine which polymers at which drug loads are going to be physically stable, and we can determine which polymers at which drug loads are going to maintain the supersaturation. And so we can identify which of the polymers that are worth a spray drying. So some of the common polymers that we often see success with, so some of the cellulosic polymers, so HPMC, the different grades of HPMCAS. We also have neutral polymers such as PVP or PVPVA64. And then some of the methacrylate-based polymers such as Udrogit L155. So an enteric polymer similar to HPMCAS. As well as looking for good solubility of the API in the solvent system, we also need to have good solubility of the polymer in there as well. So we're also looking at 2 to 5% solubility of the polymer in the solvent system. An important consideration is the viscosity of the resultant solution. So high concentrations of polymers will often lead to higher viscosity solutions. And the higher the viscosity, there may be issues around atomization of the solution when we spray dry. There may be risks with beading of material on the nozzle and clogging. And then we're also aiming typically for a total solids content in the solution of around 10%, which will help us with the efficiency of the manufacturing process. So that gives us the initial leads that we would be considering in the lab. So through preparing some of those through small-scale spray drying and characterizing the solid dispersions, we'll select which of the solid spray dry dispersions we're going to be scaling up for phase 1 clinical supply. So we'll be going from initially lab scale spray drying, so for instance using the small scale spray dryer on the top left, and at that stage we'll be working mainly to optimize the spray solution for solids content and to confirm the composition of the solid dispersion. As we move to larger scale spray drying, so bottom left we have our PSD1 spray dryer that we have in Nottingham, and we have approaches that are API sparing to establish the manufacturing process on the PSD1. So at the lab scale we would determine the solids content, and then through the use of a single spray solution preparation, we would look at exploring different process parameters such as the inlet and outlet temperature, the choice of the nozzle, the atomization pressure, the feed rate, and the interrelationship between these parameters. So this would give us an idea of the processing parameters that we need to be operating with for these initial clinical manufactures. Some of the key considerations that we're aiming for here, so because typically API is in limited supply, we want to make good use of that API. So we want to have a clinical manufacturing process that has a good yield and that we're losing little material to the fines. Additionally, as we transfer from the lab to the clinical scale, we also need to consider the secondary drying technique. So how do we drive off the solvents down to acceptable levels for the ICH limits? And we may be switching there from a vacuum oven suitable for small scale work in the lab through to perhaps a rotary vacuum dryer, which can provide a much quicker drying time course. And then, when we have these processes established, we can then transfer we can then complete the manufacture for the clinical supply and proceed with the development of the finished products for the clinical program. So that's covered some of the initial considerations during the early phase. I'll now hand over to my colleague Thomas, who will talk about the later phase considerations.
Thanks, Matt. As we transition from the early phase clinical manufacturers into late phase with an eye on commercialization, 3 common goals tend to crop up with project teams. The obvious one of these is we need to increase production capacity. Along with that often comes the need to optimize your critical quality attributes to make the best finished product dosage that we can. And along the way, as we get ready for increased regulatory scrutiny, we need to find and manage sources of risk within our product and process. 3 ways that we look at doing this are to use science and experience-driven scale-up methodologies. This is useful for scale-up, and de-risking scale-up, and for having better troubleshooting when things do go wrong during manufacture. At this stage, we also need to start linking material attributes and process parameters directly to the product quality attributes we desire. And throughout all of this, we should be doing risk assessments with the whole project team, analytical, QC, operations, and development to find and manage those sources of risk. Throughout this, I think it's helpful to have an understanding of the scale of dryers we're looking at. Early feasibility studies are often done with rather small dryers. These are your Bukis or your Proceps. The gas flow is slow, and they're typically run in open loop, which means we don't recycle the nitrogen during spray drying. We have drier processes overall. These are also done with two fluid nozzles, where we have limited particle size tunability. You'll often have very fine particles, and you need to be careful to ensure those are dry before they impact on the surface. As we move into phase 1 and pilot scale batches, it's often a time where we transition into closed loop recycle nitrogen. So we need to keep an eye on how that impacts the relative saturation of solvents at our dryer outlet. And we're also switching to high pressure nozzles, so we have to be careful about how we select that nozzle to ensure we're creating droplets of the right size for the particles that we want. Late phase, we're often looking at switching to production or even very large capacity scale spray dryers. These dryers have large drying volumes, which do allow us to have a little more flexibility with the way we're using our nozzle to create droplets that are larger and dry slower because they have more time until they impact onto the wall of the spray dryer or outlet piping. I think it's important to note that spray drying processes done at that feasibility scale are producing powders that are highly representative of what you're producing at those larger scales. Physical chemical properties of those powders are going to match. Things like your TG, which is important for the development of oral dispersion products, are going to match if your formulation matches. Dryer scales from that feasibility all the way to the large capacity cover about 2.5 orders of magnitude, which is a huge change in process capacity, which leads us to needing to use really good techniques for that scale up. I bucket scale up into two general categories. We have what I would consider solvable thermodynamics. These are things like your outlet saturation, your inlet and outlet processing temperatures, on average the residence time of a droplet from atomization until it's collected into your cyclone, and if you're running a closed loop system, the composition of that recycled gas. These can be solved and matched from early phase, early clinical, and late clinical commercial readiness development. Other things you need to consider during scale up and troubleshooting are more experience and empirical based. These are things like what size is the droplet atomized to? How efficient is your cyclone? Are you collecting 50% of your powder or 95% of your powder? And then things like sticking in the dryer. Do you have a lot of buildup on the chamber walls? And do you have good flowability of your powder for downstream processing? Another way to think about this is the links between your material attributes and your process parameters and how those will affect the product attributes of your material. Formulators and process engineers should have a mental map of the way that the variations in these inputs will change the outputs of your product. A good example is crystallinity. For spray-dried dispersion, we often want to ensure that the API is completely amorphous. This can be controlled by understanding your polymer selection and the API solubility in your solvent blend for your material attributes. And then critical process parameters are going to be things that control the kinetics of drying, which are often summarized as the outlet saturation and the outlet temperature. Another great example is the particle size. This attribute will control things like your dissolution and your flowability. This can be impacted by polymer selection on the material attribute side and process parameters, similar drying kinetic ones, and then considerations on the solids content in that spray solution and your nozzle selection. An important one that I think is missed during late phase development is excipient variability. As we're looking for late phase and commercial development, we often need to evaluate secondary suppliers for our materials. And with here, grades of excipients between vendors are not exactly the same, so studies should be taken to understand how changes in excipients impact our product attributes. Late phase development and troubleshooting, we often need to understand at a really detailed level the way that process parameters impact our product attributes. During early phase, this is accomplished with rules of thumb. Over the experience of spray drying, you learn, and with thermodynamics, understand how changes in process parameters are expected to impact your product attributes. As you develop, you get more product-specific observations from batch to batch. Those scientists and engineers are making recordings that are important for us to understand. For this specific product, how does outlet temperature impact the crystallinity or flowability of this material? And then at very late phase, we'll begin to do formal DOEs to directly link parameters to product attributes. And the goal is to get outputs like the equation shown on the bottom of this slide, where you have polynomial expressions linking your process parameters to your desired product attributes, and you'll experimentally determine those coefficients, alpha or beta in those examples, through DOEs. Another tool that we often use is risk assessments, and there's two key uses of these risk assessments. They help us prioritize where to invest our resources, and they can be used as documentation for a quick reference during troubleshooting. This helps both the operations and development team communicate clearly what to look for during these scale-up batches. The most basic form of risk assessment I like to use is a simple risk ranking, where you look at the severity of a risk and the likelihood of it happening, and that gives you a risk level, which can be used to prioritize where to spend time and how much control we need to have over ensuring that risk doesn't happen. Some high-risk examples would be things like API degradation in solution. This could cause out-of-spec product which couldn't be released, so we need to have a high degree of control over things like this. A lower impact example would be low cyclone efficiency. The severity of this is usually low, provided that we can still meet the clinical supply, and the likelihood is medium. Usually at this phase, we have a good understanding of what is driving our yields, and we can control it. So in this example, we would spend more of our effort making sure our API doesn't degrade in solution versus worrying about our cyclone efficiencies. Another useful tool during troubleshooting is to create these sorts of troubleshooting guides for project teams and the operations team, where we can have issues, causes, diagnostics, and solutions for these potential issues. So going back to our API degradation and solution example, what are some causes of this? We could have, from early phase, selected a solvent where there is some slight incompatibility with the API. Perhaps our API is sensitive to hydrolysis reactions, and a solvent blend was selected that contains water. This could have been done at early phase because it was the best option we had available at the time to get the solubility we needed, but it does expose the project to the risk of that hydrolysis reaction. Another couple of potential causes are perhaps as we scale up, there was a change in how the API is exposed to light or oxygen. Some useful diagnostics for this are clearly our HPLC solution assay and related substances methods. And then we should give our operators and scientists instructions to look for color changes in the solution. That's often a very early sign that something is going on in your solution. Some ways to address this are really formal solution stability studies. These should be performed throughout product development as we change scales and change the time and conditions we're holding our solution in. Another thing that's often useful to consider for late phase development is changing the way you hold your solution, perhaps looking at holding the solution at 2 to 8C versus room temperature, particularly if you anticipate long spray drying times and need to be covered against potentially weeks of API in the solution state. Another common set of issues is material processability. Spray-dried powders are known in the industry to have issues with flowability, and that product attribute of the spray-dried intermediate needs to be controlled and troubleshot during scale-up. This poor flowability comes from the generally low bulk density of these materials and the small particle size. The diagnostic of this is physical characterization and then downstream experience. You need your spray drying team to be in communication with the team developing the solid oral dosage form to understand how impactful this poor flowability is on their ability to produce the material. Some ways we can address this with the spray drying process is to optimize our atomization to create larger droplets, changes in the dryer settings to create denser material, and looking at improving our spray drying solution and perhaps increasing the solids loading if we can. Some equipment-driven troubleshooting issues is that low cyclone efficiency. This can be caused by an improperly sized cyclone. Perhaps we're pushing process throughput and we're now operating in a regime that the cyclone wasn't originally designed for. Perhaps we're creating a lot of fine powder. Or one that often creeps up is if you have material sticking on the wall of your chamber, and then if your pneumatic hammers knock that material off, you can clog or overload your cyclone. Some ways to diagnose this are ensuring you check the design calculations of your cyclone, creating larger droplets to create larger particle sizes. And if you see cyclone clogging, the most common diagnostic for this is you'll have collection container yields of 90%, 90%, and then suddenly you'll have a collection container yield of 50%. This can often be diagnosed by also pressure monitoring around your cyclone. If that pressure drop changes, you know that something has changed in the physics of that cyclone. Now I'm going to walk through a specific case of some troubleshooting we saw during tech transfer of a batch from the PSD1 scale to the PSD2 scale, where we had no historical observations of crystallinity in our spray-dried powder until this batch. There was a few initial hypotheses on what had happened. The first one was incomplete API dissolution. An issue that we have with this hypothesis is that it's really difficult to assess post-manufacture. Once that material's been sprayed, we can't go back in time and assess if it was fully dissolved in the solution. And a few things we had in place to de-risk that were visual confirmation of dissolution. The operators observe the processing tank and ensure that it's a clear solution, and a minimum mix time was in place. The other hypothesis that was perhaps during this scale-up, we changed the drying kinetics of those droplets. This wasn't likely, though, because we had a thermodynamically scaled process and consistent particle size, meaning likely consistent droplet size between the batches. What I will draw your attention to is at the bottom of the table, we had a fairly large change in batch size and solution size, which had led to what we eventually identified as the root cause of the issue was a change in our collection container size. Historically, on the smaller scale dryer, we had been using 10-liter collection containers and changing them out every 3 hours. At the larger scale, we were using 100-liter collection containers and changing them out every 7 hours. This change in size and then separation from the process by double flap valves led to a change in temperature in the collection container. This change in temperature actually leads to an increase in the relative saturation of those solvent vapors, being the water and the acetone, which is known to plasticize the spray-dried dispersion. So our materials in the collection container for our tech transfer batch would have had a much lower TG relative to what we had seen historically. That lower TG leads to plasticity, which can lead to API crystallization. The ways we mitigated this after we had identified this as the root cause were to reduce our container size, increase the frequency at which we change those out, and then a major one was the implementation of a dry nitrogen sweep. So you can sweep the headspace of that collection container to remove that solvent vapor so that you're not plasticizing your SDD as it sits in that container. An additional one that we assessed and didn't actually move forward with was reformulation. I think it's important during late phase troubleshooting to understand that there are things we can change about the formulation and the process. Particularly in this case, we were looking at reducing the amount of water in the spray solvent, as that's often the material that's most sensitive to changes in temperature, changing the relative saturation in your outlet and collection containers. So through all of this, I really like to link back to that scale-up and troubleshooting needs to be science and experience-based. We have available to us tools that solve the thermodynamics of the spray drying process and a load of experience for how to scale up these processes from feasibility to early clinical and then get ready for commercial readiness. That's part of our offering at Catalent for our end-to-end spray drying technology. At the development stages, we have full on-site analytical characterization and method development. These teams often do our pre-formulation testing where we select which polymers and solvents are best for the spray drying process, and then transfer that to our process development team to get the initial spray dried powders available for in vivo studies in animals and clinical readiness. We have the capability to process with organic and aqueous solvents and potent and non-potent molecules. We have sites that are specialized in R&D and GMP manufacturing, covering that early feasibility through phase 1 to 2A of development. As part of our integrated surface offering, we also have scale-up and commercialization, where we have in-house formulation and process expertise as we look towards those large production capacity dryers. And having that in-house allows us to have more direct communication between the early development teams and late-scale development teams. For GMP manufacturing, really looking at that phase 2B through commercial readiness. These sites do have scales of dryers appropriate for those low-volume orphan and pediatric products, all the way to large-scale blockbuster products. Thank you for your time and attention during this section. With this, I'll hand it off to our colleague, Carolina Corzo.
Thank you very much, Thomas and Matt, for a really nice talk. I will now land on a specific application of spray drying for lipid-based formulations. Based on our experience at RCP, I will walk you through the challenges and general limitations when spray drying lipids. And of course, at the end, I'll provide some solutions and a strategy of how to overcome this. Before jumping actually to science, let me give you a couple of words about RCP. RCP stands for Research Center Pharmaceutical Engineering. We are a non-university independent research company in the field of pharma, process, and product development. We are located in Austria, in the city of Graz. Currently, we are 150 employees coming from more than 20 different countries, so a very international team that we are. What we do is to support pharma industry in understanding process and product design at early stage drug product development. And we do this through service projects and long-term collaboration with pharma industry. RCP is divided in 4 areas, as you can see here, and covering basically the whole spectrum of product development. And so far, we are collaborating with more than 25 scientific partners among universities and research centers, and also more than 130 industrial partners, among them some big pharma players. And for my talk today, I will first give you some generalities about spray drying of lipids, what we know so far, especially hurdles and limitations. Then I will walk you through some rational process design for lipids, for spray drying of lipids. And basically, at the end, I will just give you some examples of how successful these strategies can be with some case studies. So let's start with spray drying of lipids, some hurdles. First of all, let's understand why we would like to spray dry lipid-based formulations. Well, lipids as excipients can be very beneficial materials for drug delivery. They are coming from natural sources, and because of that, they offer a very good biocompatibility and low toxicity in the human body. Of course, they can provide as well different functionalities to the final dosage form, such as enhanced drug bioavailability down to taste masking, for example. The problem comes when we want to spray dry lipids. These materials are often very complicated for spray drying. They are characterized by extremely low yield, even below 20%, well deposition, sticky products, and many of the issues that Thomas mentioned already are very much expensive for lipids. So to this point, you will wonder then why we would use spray drying for lipids if it's quite complicated. Well, the answer is quite short and straightforward. By spray drying lipids, we can provide effective particle engineering for inhalation products. And this can vastly improve the performance of conventional and inhalation products. And I will go a bit into details in this point, lipids for inhalation. Well, we are talking about providing dry powders for inhalation or DPIs, which are free of large carrier particles. They are called like carrier-free DPIs. And compared to conventional carrier-based DPIs, they can offer a lot of advantages for the patients. So if we take a look down here to the behavior or the performance of conventional carrier-based DPIs, they are composed basically by micronized drugs, which are then blended with a carrier with a larger particle size, basically lactose, which is the most common carrier for DPIs. This blending step is made in order to provide a better flowability of the product and better aerosolization performance. Then the DPI is inhaled by the patient, and in this aerosolization process, there should be a detachment stage in which the large carrier particles are basically separated from the micronized drug. The idea is for the drug to reach lung deposition and to actually reach the stage of the lung that we want to target. However, so far, the performance of these conventional carrier-based DPIs is not optimized. It is known that from 40 to 85% of the dose is not delivered to the site of action. This is either due to it staying in the inhaler, in the device, or it's simply not properly detached, meaning that the micronized drug will stay in the upper tract and it will be simply swallowed. So at the end, an average of 40% of the drug is actually reaching the site of action. How can lipids help in that by providing carrier-free DPIs? Well, first of all, we can avoid the blending step, and we provide particles that are readily inhalable as lipid microparticles, which are produced by a spray drying, of course. In this sense, we can provide a modification of the drug crystallization into smaller particles, which, as I said, can be inhalable directly without a blending step. And also, lipids are known to enrich the surface of the particle during particle formation by spray drying, and it is associated with enhanced aerosolization and, of course, a better performance of the DPI. Also, as you know, by spray drying, in general, we can provide many different attributes such as morphology, sizes, and so on, which will help us to target different phases in the lung. And all of that will actually enhance the lung deposition and to have a better performance for the patient. And I think so far I gave you enough reasons to actually face this limited processability of lipids by spray drying and to understand why and to try to give some rational solutions. Why the processability is so limited? Well, there is one main reason, which is the low melting temperature of lipids. This is a problem in relation to the outlet temperature during spray drying. Although this is the most important reason, this is not the only one, unfortunately. Lipids are also complex materials which are composed by multiple phases, most of them having different melting temperatures, which can create phase separation during spray drying. Also, other lipid crystallization pathways such as polymorphism may affect processability by spray drying. Anyways, the lipids have been used in the industry, and the solutions that are given so far is to include some additional solid formers in the feedstock for spray drying, meaning salts or sugars or polymers are mixed in the solution for spray drying, and this, of course, increases the processability of lipids. However, there are disadvantages such as dose dilution. We cannot reach the highest dose possible because of the dilution effect of extra excipients, and of course, more complex formulation development with more materials included. What we want to show you today is how a rational process design combining critical material attributes with the process parameters engaged with the process performance can actually give us a simple formulation completely based on lipids with a high yield. So let's go to this rational process design. The strategy that we propose might seem simple. It's first of all to select lipids with the highest melting point possible. This will facilitate the process a lot. Then to run a pre-formulation step in which we check any kind of interaction, drug or solvent with the lipids that might impact the melting point of the formulation. And of course, to inspect any other transition. And based on these findings, we can fix a boundary for the outlet temperature of the spray dryer. The question is so far here, okay, this strategy looks quite simple to follow, but is that enough? Or the more direct question would be, is that applicable for any kind of lipid? Well, let's see now. I will show you some case studies where we will prove this concept. In these case studies, what I want to show you are two different particle attributes that are supposed to provide different drug delivery applications. So with that, we want to provide totally lipid-based formulations, of course, no additional excipients. And the two examples that I have here are, the first one is for the systemic delivery of analgesics. For this part, we need DPI, a dry powder that is dispersible without the addition of carrier particles that is inhalable, meaning that having an aerodynamic size between 1 and 5 micrometers. And when the particles are finally deposited, they are expected to diminish clearance by macrophages as much as possible in order to deliver the drugs via systemic absorption. And in order to provide such characteristics to the final product or such product attributes, we fix these particle attributes, as you can see here in the slide, via systemic delivery. And we want to provide these particle attributes via spray drying. On the other side, we have another example, another case study, in this case for targeted delivery to alveolar macrophages. Here we have slightly different particle attributes, and the most important difference is that the particle after deposition should be enhancing or triggering the phagocytosis in order to have a targeted delivery inside the cell and deliver the drug inside. So for that, these two examples are the application of lipid-based particle engineering, I would call it like this. For feedstock, for spray drying, we will prepare the systemic delivery formulation by an organic solvent solution, and the other formulation by an aqueous suspension. And this is quite a kind of a random selection in order to show you how wide the application of lipids can be for different type of spray drying processes. I will mostly focus on the systemic delivery formulation. I will show you more details about it, and all the findings can be directly applied to the second case study, for which I will give you just the short summary. So for the solution-based spray drying, as I said before, the selection of lipids with a high melting temperature is very important, and I wrote here on purpose relatively high melting point because lipids in general don't provide so high melting point. So we are talking here about the polyglycerol esters of fatty acids, for example, which are materials with several glycerol units attached to each other. As you can see here in this example, either PG2, 3, or 4 molecules, and then they are esterified by behenic acid. That's why they are called behenates. And this structure provides a relatively high melting point of around 74, 75 degrees. I'm having here also an example of a PGSA in light blue with a relatively low melting point. This is a comparison of how important it is to provide a relatively high melting point on lipids. The other materials are glycerol dibehenates, tripalmitin, and behenoyl polyoxyl glycerides, which are also having high melting points, and as a model drug, ibuprofen. For the second part, as you remember, the pre-formulation step was quite important to carry out, and it's a quite easy and fast step. In this case, since we are spraying a solution, a solvent casting pre-formulation will give us a good idea on a prediction of the solid state outcome from spray drying. So from here, basically, we prepare solutions containing ibuprofen and one lipid, and ibuprofen in different percentages in respect to the lipid. Then the solvent was evaporated and the remaining solid is analyzed by a DSC. I'll show you here one example of these findings for PG3, one of the PGFAs that I show you in combination with ibuprofen. So in the x-axis, we can see the percentage of ibuprofen in relation with the lipids, and in the y-axis, we can see the melting temperature. So the very clear observation here, the first one, is that even though it shows lipids with high melting point, like around 74 degrees, any kind of interaction with ibuprofen will immediately drop the melting point of the mixture. And what we are seeing here is a eutectic interactions among these two components, which is characterized by melting point depression. And what we see here is that now the real melting point of the formulation is not actually the matrix, 74 degrees, but the eutectic temperature, which is quite lower than 60 degrees. So very first observation is, okay, we need to provide an outlet temperature sufficiently below the new melting temperature of the formulation. And how is sufficiently below? We will see in the next slide what is the recommendation on that part. Important is also to mention that with lipids, and especially with this system, what we will provide as a product is a crystalline dispersion. We've been talking about amorphous solid dispersions in the previous talks, but in this case, we are providing crystalline crystals of ibuprofen dispersing crystals of lipids, so a simple crystalline dispersion. We do the same with each lipid. We have here the phase diagram in combination with ibuprofen. I won't go through many details, but just to point out that everything which is in light blue, as you remember I mentioned, we chose this lipid on purpose to show how a low melting point may affect the process. And yeah, the melting point of the original material is low, and now in combination with ibuprofen is of course even lower, down to 45 degrees, sorry. The other lipids are either showing multiphasic systems like the one you can see in BPG ibuprofen, for which many liquid and solid regions are mixed, and this will provide a risk of phase separation during spray drying. The rest of the lipids, as you can see, are having only one melting temperature, the eutectic temperature of around 60, 63 degrees more or less, which might be still processable by a spray drying. So we use these temperatures here, the eutectics, to provide a boundary, a rational boundary for a critical outlet temperature. And based on this, we can, of course, go to the next part, which is to define the process parameters. Okay, important is to know what's the boundary for the outlet temperature, but the most important part is actually to provide the target particle attributes that we defined previously in the quality target product profile. So for that, we can consider the Peclet number. For that is very, very useful. And I think until this part, I don't need to go into details. Thomas explained very clearly how the process parameters will immediately impact the final product attributes. So here it's a matter of screening the drying kinetics, the matching heat transfers, and so on, especially considering the atomization part, for which we should provide a small initial droplet size after atomization, because remember we are working with inhalation products. So based on all of this, and of course considering the boundary outlet, we fix some process parameters, and I won't show here all of them, but important to mention the inlet of 71%, and in this slide I want to show you the process performance of each material. So let's go step by step. I have here two figures. I hope you can see them clearly. In the first figure, we have a comparison of the temperature and the yield. So down in the y-axis, we have the name of each lipid in combination with ibuprofen, and we have in the orange points the outlet temperature of the system. In green, the green points, we have the melting point of the pre-formulation that we defined previously, and the blue bars are the process yield. In the other figure, We have droplet size, the initial droplet size after atomization, and the particle size of the product at the outlet. So we are comparing these two to provide let's say, a quantitative idea of the agglomeration at the outlet. So when the particle size is actually larger than the droplet size, we can have a sign of agglomeration in the system. Usually it's like that. Of course, there are some conditions in which larger, very low density particles are provided. This is not our case here. So this statement of particle size larger than droplet size will give us a sign of agglomeration. So what we are seeing here, first thing is that for all solutions, we are providing an outlet temperature of around 35 degrees. And actually, for the two lipids that I show you with the very low melting point, we have a very close distance between the melting and the outlet temperature, which will immediately provide a very low yield, even below 20%, and particle agglomeration, as you can see in the figures. On the other hand, lipids which are having a melting temperature far away from the outlet temperature provided yields even above 80% and completely no agglomeration in the system. So the very first clear conclusion is that at least a difference between 20 degrees should be given between the outlet and the, let's call it, the real melting point of the formulation in order to increase the yield and provide less agglomeration. This is clear, and that can also be applied for other kind of materials such as sugars, considering the glass transition and so on. But for lipids, we can establish as at least 20 degrees should be given. Unfortunately, as you can see here, this is not applicable for all lipids. This is not enough, considering the difference between outlet and melting is unfortunately not enough for all type of lipids. In this case, for tripalmitin, we are seeing here exactly the same thermal conditions. Difference between outlet and melting are the same. However, the yield is extremely low, even below 10%, and there is agglomeration in the system. So consideration of the outlet is not enough for some lipid systems. How can we explain this, and how we can actually give solutions for this? Well, if we consider a simulation of the droplet and particle pathway inside the drying chamber, please now take a look to pathway number 1. Okay, and this is the most straightforward and simple trajectory of a droplet particle. So if we imagine on the inlet of the dryer the formation of a droplet, which is immediately exposed to the hot drying gas at 71 degrees, then the droplet will rapidly go down until eventually the solvent is completely evaporated and a solid particle is formed. This solid particle will just follow this center line very nicely and go directly to the outlet, and then it will be exposed to 35 degrees, which is the outlet temperature. We shouldn't forget here that when the droplet is still existing as a droplet, meaning that the solvent is in evaporation, it will never reach 71 degrees because the saturation temperature of the mix of the vapor and the gas will be lower than that, let's say around the wet bulb temperature of this mixture. So it will never reach 71. In this case, the particle will be only exposed to the highest 35 degrees. On the other side, not all the particles, unfortunately, are following this ideal pathway, but they can follow also pathway number 2, which is after particle is formed, it can be entrapped in this kind of recirculating eddy, which eventually will bring the particle to the inlet without having the evaporative cooling effect during solvent evaporation, so it will actually get exposed to 71 degrees. What happens with these particles actually? What happens when they are exposed to such high temperature? Here we have a DSC analysis of PG3 with the highest yield and tripalmitin, that you remember, very, very low yield. So all the particles reaching the inlet are going to be molten immediately, since the melting point is exactly below that. So all of them are molten. The difference between PG3 and 3-palmitin is, however, the recrystallization temperature of them. So after the particle is exposed and molten at this higher inlet temperature, it will almost immediately get recrystallized, and it will have a very short residence time in the chamber in a molten state, meaning that wall deposition and product loss will be actually neglected. In the case of 3-palmitin, the recrystallization is even very further away from the melting, as you can see. So the existence of molten fractions in the drying is unfortunately longer. Also, partially, a fraction of these particles will stay partially molten in the outlet, which will also increase the agglomeration of the system. So basically, a long residence time of molten fractions in the drying chamber will cause wall deposition in lipids, and these soft fractions in the product vessel will cause agglomeration and, of course, impact the yield. I want to point out here that it's quite easy screening to use the DSC with a pre-formulation and to take a look to melting temperature, crystallization, and so on in order to choose correctly the inlet and the outlet temperatures. And I just want to explain you why they have such a different behavior between melting and crystallization. Actually, the answer is very short. 3-palmitin suffer polymorphism, meaning that the beta form with the highest melting point will be molten in the inlet, and then it will try to recrystallize in the alpha form, which has a low melting point and a low crystallization temperature. And that's why we have such a problematic process. On the other hand, PG3 doesn't have any kind of polymorphism, so melting and crystallization temperatures are very close to each other. And finally, we can use this simple DSC analysis to predict some issues that might happen, like how close the recrystallization is to the outlet, or if there are existence of low recrystallizing fractions in the material. And with that, to settle down a little bit all the knowledge that we gathered so far, what are the critical considerations when spray drying lipids? Well, first of all, definitely select lipids with highest melting point possible. This will give us an, let's say, a wider, a broader design space for spray drying. Run a fast and easy pre-formulation step. As you could see, this pre-formulation information gave us actually a lot regarding the prediction of solid state outcome and certain problems that we might face. If we find there is a melting point depression or polymorphism, we have to set the outlet temperature at least 20 to 25 degrees below the expected melting or recrystallization, and for polymorphic lipids or multiple phases, it is better to avoid melting in the system, meaning that setting the inlet below the melting will be the ideal situation. If this is not possible, because dry modeling is not sufficient or simple particle attributes are not matching these conditions, then we need to fix the outlet way below the recrystallization at least. (20 to 25 degrees) in order to improve a little bit the processability of such lipids. And just with this, I just show you quickly what is the performance of these particles with PG3 and ibuprofen. You can see here the performance as a carrier-free dry powder for inhalation. We use a simple inhaler and aerolyzer, and we use NGI for this evaluation. The emitted fraction of the product is quite high, 90%, meaning that only 10% of the product is staying in the inhaler, which is quite a nice number compared to carrier-based formulations. This is actually associated to surface corrugation in the particles and the lipidic composition on the surface, and this was actually provided by a high surface mobility during drying because of the Peclet number above 1 and also a low solid content of the feedstock, which was actually 1.5% only. Inhalable particles were provided with MMID of 3.5 micrometers, high fine particle fraction 45%. If you remember, for carrier-based DPIs, we reach around 40% only, and yeah, such a nice performance in general. And applying exactly the same knowledge to different targeted particle attributes, we could also generate a yield of 80% using an aqueous suspension, in this case with a Peclet number below 1, and with slightly different particle attributes to target alveolar macrophages and with, again, a high fine particle fraction of 59%. And yes, with this, all of these recommendations, we could actually provide completely lipid-based particle engineering without a lot of complications. And thank you very much for your attention, and thank you also for all the institutions that are supporting this work, and to Catalent for this opportunity. Thank you.
Fantastic. Thank you for sharing your insights in this presentation. Now, before we move to the Q&A, for the folks who are watching, if you haven't already, you can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. All right, let's move into the Q&A, which the first question is for Carolina. Are there inhalation products in the market based on lipids and spray drying? And if yes, how have they managed to provide an efficient or cost-effective manufacturing process? Also, is there a way to increase the yield even more?
Thank you for this question, very important. Yes, there are actually inhalation products in the market based on lipids, basically based on phospholipids, which have a higher melting point but different transition temperatures, which also make the process a bit complicated. They managed to be in the market so far because the feedstock, as I was explaining before, is a combination not only of lipids but also sugars or salts in the system. So the dose is quite reduced, I would say, but this definitely helps to increase the yield of particles. How can we increase the yield even more? Well, definitely considering cyclones with higher performance can be done actually for any kind of materials, not only lipids, and to use also bag filters can increase also recovery of fines if this is one of the issues. But for lipids, I would say just setting the temperatures according to the transitions, polymorphic transitions or melting points, whatever kind of transition, in order to increase as much as possible the yield.
Fantastic. Thank you so much. The next question is for Tom. How does particle size change from lab scale to pilot and production scale?
Yeah, that's a great question. I'll flip back to our slide on the spray dryer scale comparisons. So at the lab scale, we're typically using these small chamber dryers with 2 fluid nozzles. And this really limits us to slightly smaller particle sizes than we typically want for downstream flowability. As we switch into that pilot scale with that increased chamber size and switching to a high pressure nozzle, we're able to increase that particle size into the target range. As we move from pilot to production, at this point we're usually not trying to further increase the particle size significantly. We're trying to match the dissolution and flowability performance that we got at the pilot scale.
Great. The next question is for Matt, and that is, are there any limitations on the choice of solvents for spray drying?
So I think some of the limitations I covered earlier. So we need to think carefully about the ability to remove the solvent down to ICH levels. So really having the ability to get it properly dry is a very important consideration. There are also a few solvents that don't have great compatibility with some of the equipment. So we often see problems with the likes of THF, which is often a great solvent in terms of solubility, but it does often cause problems for the seals on the equipment. The other constraint is the boiling point of the solvent. Though we have been exploring some of the more highly boiling but really great solvents like DMSO and DMF, and have had some success in employing those in at least small-scale spray drying.
Fantastic. Next question is for Carolina, and that is spray drying of lipids applicable to other materials that are also complex in their solid state?
Yes, actually all the steps that we went through, pre-formulation and analysis of DSE basically, can be immediately applied to other kind of materials. Maybe they won't have polymorphic transitions or multiphasic crystallization, but we can talk about also glass transitions that might also impact the processability of any material. So yes, everything that we saw so far can be applicable to any other kind of material. Yeah.
Great. The next question is for Tom, and that is, when should a project team start using scale-up risk assessments?
Risk assessment should really be used at every stage of product development. As we start with preclinical feasibility with an eye on phase 1, we're pretty much scaling up constantly throughout that process. And then the rigor and formality of these risk assessments will increase as we move from those early phase, phase 1 considerations to late phase, but a risk management-based approach to development will really be key for a project team to know how to allocate their limited resources to the highest impact problem statements.
Okay, great. The next question is for Carolina, and that is, are the lipid transitions during spray drying somehow avoidable?
Yeah, unfortunately, no. Lipids are in general complex materials in all senses, transitions, polymorphic transformations, and they can not be avoided, but they can be somehow controlled using these pre-formulation tools, like how it's the impact, for example, of the drug loading in the transition of the lipids can trigger the most stable form of the lipid, for example, and with that we can kind of control the transition in a way, but it's unfortunately not avoidable. Yeah, even though lipids are very complex molecules in the solid state and crystallization, they kind of balance this with all the benefits that they have, natural materials and very low toxicity and actually very functional materials, so it's a balance of a combination of these two, but we can kind of control this transition. Yeah.
Great, thank you. Next question is for Tom, and that is, what is the expected impact on product attributes when transitioning from open to closed loop drying gas?
That's a really great question. I think there's two approaches I've seen used for making that transition. The first one is to slightly decrease that solution feed rate to match the drier outlet saturation between the open loop and closed loop processes, accounting for that nitrogen containing some non-condensed solvent vapor when you're recycling it. By matching that outlet saturation, this will really minimize changes in the way the droplets dry in the spray drying chamber, which will help you match product attributes like your particle morphology and flowability. Another thing you can do, though, the other approach, is to actually allow that recycled solvent vapor to slightly increase the outlet saturation. This will slightly slow down your droplet drying and can transition you from those inflated balloon SDD particles to the more collapsed raisins, which often have improved density and flowability. So during late phase scale-up, this can be something that can be assessed as a way to improve that quality attribute of the flowability of your material.
Great. The next question is for Matt, and that is, how long can the API solubility be to still have a viable spray drying